Good evening, everyone, and welcome back once again to yet another super cool and supremely amazing one shot revision crash course for NEET 2021 on the chapter Cell, the basic unit of life. So today I am coming in front of you with a supremely amazing one-shot revision series of the chapter in which we are going to have an overall synopsis of the chapter Cell, the unit of life, where we would be discussing the important topic which can come as a question for our NEET examination. So without wasting the time, let's get started with the Cell Unit of Life crash course revision for NEET 2021. Those who are watching the session right now, don't forget to hit the like button, do share it with your friends and subscribe this amazing platform where you can clarify your doubts. You can have the quizzes on our amazing Telegram group. The details of the Telegram group for those who are watching the session brand newly will be introduced soon. So let's get started with this chapter, Cell, the unit of life. So in this chapter, first of all, we are coming with the cell theory. We are coming with the cell theory, but before let's discussing about the cell theory, I would like to tell you about two important discovery. The first one is the Robert Hooke. The first discovery is all about Robert Hooke. What was the discovery of Robert Hooke? Robert Hooke was the first person to observe a dead cell through the microscope. Okay, Robert Hooke was the scientist who discovered the cell. He observed a dead cell of a cork. Through a cork, he observed the dead cell. So he was the first person to observe or to discover the cell. But much before he introduced the cell, or he coined the term cell, Anton van Leeuwenhoek already saw the cells under the simple microscope which was prepared by himself, and he observed the living cell. He observed the living cells. But instead of calling it as the term cell, he called them, or he called those minute substances or particles as corpuscles. He undergone, or he took out his experiment through different materials. For example, sperm was one of his experimental material. Second one is bacteria, sperm. All those things were different experimental materials which were used by Anton van Leeuwenhoek. And one more important point which you have to be aware about when you are dealing with this chapter is about the scientist known as Robert Brown. About the scientist known as Robert Brown. Robert Brown was a scientist who discovered the nucleus. Okay. Robert Brown was a scientist who discovered the nucleus. What is nucleus? The control center of the cell is the nucleus, and he discovered the nucleus in the year 1831. Okay, Robert Brown was the first person to discover nucleus inside the cell. He discovered nucleus in the year 1831, which is a previous year MCQ question. Who observed the dead cell and who observed the living cell? Previous year MCQ question. So let me raise this and let's get into the chapter Cell, the unit of life. First of all, let me discuss about the cell theory. What is actually cell theory? Those certain postulates which was proposed by different scientists. The first of all, the cell theory was proposed by two scientists, namely Matthias Jacob Schleiden and Theodor Schwann. Okay, Matthias Jacob Schleiden, who was a German botanist. This is a previous year AIMS question. Both the countries of these scientists. Schleiden was from Germany. He was a German botanist. Okay, guys, you have to be very careful about it. He was a German botanist, and Theodor Schwann was a British zoologist. Okay, Theodor Schwann was a British zoologist. So definitely you know what were their proposals or what were their postulates. He told that all plant cells are made up of whole plants, all plants are made up of structural basic functional unit known as cells, because he was a botanist. Right, everyone? He was a botanist, so he proposed that all plants are made up of single basic unit, which is known as the cells. But what did Theodor Schwann, who was a British zoologist, propose? That is, all animals were made up of cells. So, since he was a botanist, he proposed that all plants were made up of. Since he was a botanist, he proposed that all plants were made up of cells. And since British zoologist was Theodor Schwann, he proposed that all animals are made up of cells. Okay, everyone. So the cell theory was formulated by Theodor Schwann and Theodor Schwann and Matthias Jacob Schilden, and this theory was modified by, or the theory was given its final shape, which is a previous year MCQ question. Who gave the final structure to the modern cell theory? This Rudolf Virchow is considered as the father of modern cell theory, or he was the person who formulated the cell theory modern. Okay, final formulation of cell theory was done by Rudolf Virchow. So let's discuss what were the points that were included in the cell theory. All living organisms are composed of cells and product of cells. All cells arise from pre-existing cells. NEET 2019 question. Who proposed the statement all cells arise from? NEET 2019. Who proposed that all cells were arising from pre-existing cells? This was by Rudolf Virchow. But he made a statement in his own native language, that is hominis cellulae. That means, in English, it means all cells arise from pre-existing cells, and the cells are the structural and functional unit of all living organisms. Is that clear? Sorry. Yes. First of all, we are going to discuss about the prokaryotic cell. Before we are discussing about the prokaryotic cell, let me tell you something which is super important for your exam point of view. That is, let me tell you one important point. 1 second, let me take a plain screen. Yes, let me take it out. Yes, everybody, listen. Cells can be divided into different types based upon the size and shape of the cell. So first one which you have to study is the smallest cell, which is PPLO, which is pleuropneumonia-like organisms, mycoplasma. And you have to study mycoplasma, pleuropneumonia-like organism. And the largest cell is the ostrich egg. Ostrich egg is the largest cell. The smallest cell in the human body can be either RBC, or you can call it as, or you can also write it as to be sperm. If both of them come, you can go with RBC. Okay. Then nerve cell or neuron is the longest cell. Nerve cell or neuron is the longest cell. Okay, everyone. Neuron or nerve cell. Okay, so these are some of the important points before we move on to the prokaryotic cell. So let's discuss about the prokaryotic cell. I hope whatever we are discussing is clear for each one of you over here. Yes. So modifications of the cell envelope. Modifications of the cell envelope. First of all, we are studying about it. Okay, everyone. So cell envelope consists of tightly bound three-layered structure, which is the outermost glycocalyx, followed by the cell wall in case of plant cell, or the plasma membrane. Glycocalyx is the outermost shell or the outermost layer, which may be in the form of loose sheath, called the slime layer, or the glycocalyx can be thick and tough sheath, which is called the capsule. Okay, everyone. So the glycocalyx may be a thick layer. So if it is a thick layer, then you will call them as the capsule. If it is a slimy, loose connective layer, then you will call them as the slime layer. If they are slimy in nature, you will call them as the slime layer. You have to be very important. This particular glycocalyx helps in the attachment or fixation of the bacterial cell. Sorry, I think it's not. Hope I know I have made a mistake when I read over a sentence over here. That is, we were discussing about the prokaryotic cell. I'll clear the same, my mistake from my side, I'll clear it. So prokaryotic cell means that do not have a well-defined nucleus. No wall or no well-defined nucleus. Right, everyone? But when I told over here, I told like plant cell. So I thought I just make confusing myself with this plasma membrane and cell wall. That's why the mistake came. So once again, I'll explain the same for you. So cell envelope, it consists of three-layered structure, which is the outermost glycocalyx, which is followed by the cell wall and the plasma membrane. Glycocalyx is in the form of loose sheath, which is called the slime layer. Okay. And the glycocalyx is in the form of a thick and tough sheath, which is called the capsule. Next, guys, this is the outermost layer, you suppose. No, let me draw everything and I'll tell you. Okay, just imagine what I'm right now drawing is the glycocalyx layer. Okay, what I'm right now drawing is the glycocalyx layer. And what I'm going to draw now is the plasma membrane. So the plasma membrane extends like, I'll draw with the normal pen so that the content would be more clear for you. Listen, everyone. So the plasma membrane may extend into the cytoplasm of the cell as extension. Okay, like this kind of extension we can see in the plasma membrane. So these extensions of the plasma membrane, you call them as the mesosomes. You'll call them as the mesosomes. Mesosomes are the extensions of the plasma membrane. They can be in the form of vesicle, tubules, or lamellae. What are the functions of mesosome? Previous year MCQ question. They help in cell wall formation, DNA replication, distribution to the daughter cell, respiration. The organ or the organelle, the structure which is present in the prokaryotic cell, which is similar to the mitochondria in eukaryotic cell. It is the mesosome. Very important AIMS question. Secretion, increases surface area, enzyme content, etc. Okay, everyone. So these are some of the important points which comes in this slide. Right. So let me discuss one more time. I'll telling the important points. It is a three-layered structure which consists of the outermost glycocalyx followed by the cell wall and then the cell membrane. Guys, this is a previous year question. Okay, this is a previous year question. To arrange the three layers in the correct from inner to outside or from outside to inside. Glycocalyx can be in the form of a loose sheath which is called the slime layer and in the form of a thick tough covering which is called the capsule. Next we discuss about mesosomes. Mesosomes are the extensions of plasma membrane. They can be in the form of vesicle, tubules and lamellae. They help. This whole content is very important. Okay, the whole content is very, very important, everyone. Okay, so cell wall formation, they help in cell wall formation. Then DNA replication, distribution to the daughter cell, respiration. That is why they are analogous to mitochondria in eukaryotic cell. Secretion to increase surface area, enzyme content. So all these things are important functions of mesosome. Clear, everyone? So let's move on to the next one. The next structure which we have to discuss over here is the pili and Fimbriae. Surface structures of some bacteria which helps them to attach to the streams and to host tissues. Guys, if this is an MCQ, then the answer should be Fimbriae. Okay, you have to go with the answer Fimbriae and not pili. Pili means sometimes helps in sexual conjugation or sexual reproduction. Okay, so the surface or these structures in bacterial cell or prokaryotic cell that helps them to attach to the rock surface and to the host tissues, then the answer is to be Fimbriae. Next, the genetic material is not covered by a nuclear membrane. In addition to the genomic DNA, the single chromosome or circular DNA, they have a single chromosome or a circular DNA. Many bacterium have small self-replicating double-stranded DNA which is known as the plasmid. Plasmid contains many genes like the antibiotic resistance gene, antibiotic resistance. Okay, so Fimbriae is the structure which helps, the surface structure which helps in the attachment. Okay, surface structure which helps in the attachment them to the rocks in streams and the host tissues. Next, their genetic material is not covered by nuclear envelope. They have small circular self-replicating double-stranded DNA which is known as the plasmid. Plasmid have antibiotic resistant gene which you would study in your biotechnology chapter. Is that clear? Yes. Next is all about the ribosomes. What is actually ribosome? Ribosomes in case of prokaryote, they are 70S. Associated with the plasma membrane, they are the site of protein synthesis. Polyribosome or polysome, very important. Several ribosomes may attach to a single mRNA to form a chain called polysomes or polyribosome. They translate the mRNA into protein. Previous year MCQ. Polysomes or polyribosomes. Next is inclusion bodies. Inclusion bodies stores reserve material. They lie freely in the cytoplasm without bounded by any membrane. For example, the examples previous year MCQ. Phosphate granules, cyanophysin granules, glycogen granules. They may ask you some other name of the granules. Since these three are mentioned in your NCRT, your options should also be as per that. Is that clear for everyone? Is that clear? Is it clear? So we have discussed about the cell theory. Then we discussed about the mesosomes, glycocalyx, cell wall, plasma membrane, pili, fimbriae, genetic material, ribosomes, and inclusion body. Guys, after this lecture, you will get the PDF of this particular lecture in our Telegram group, or as well as in the description box, as well as I'll provide you with the NCRT extract of this chapter, which is very, very important. So as I have already promised, you have the crash course happening right now in our Biopoint, along with the detailed lectures of the most important and the toughest chapters recommended by the students, like principles of inheritance and variation, human health and diseases, molecular basis of inheritance, biomolecules are yet to be completed. They are already started. Biomolecules, you had a lecture the previous day, right? One or the other day. Previous days you had the lecture of molecular basis of inheritance. You had one lecture of principles of inheritance. So once again, I am just making up these slides by inserting the slides and everything. That's why the second lecture is getting delayed. Okay, don't worry, I'll be coming out soon with the second part of the chapter with monohybrid cross. So stay tuned for daily updates. You have to join at t.me/neet_biopoint, where daily polls, handwritten exams, daily schedule updates, daily telegram quizzes, and mentor doubt clearance happens. Wow, is it? So join the Telegram group as fast as possible if you are not become a part, or if you have not become a part of Biopoint till now. So don't forget to join the Telegram group. Okay, everyone. So next session is all about eukaryotic cells. Eukaryotic cell possesses an organized nucleus with a nuclear envelope and have a variety of complex locomotory and cytoskeletal structures. First organelle of eukaryotic cell, which we are going to discuss over here, is the cell membrane. That is, Singer and Nicholson in the year 1972 gave the fluid mosaic model. Okay, fluid mosaic model. Let me highlight it so that it would be more clear, perfectly clear for each one of you. Okay, so they have an organized nucleus with a nuclear envelope and have a variety of complex locomotory and cytoskeletal structure. First, let's discuss about the cell membrane. The cell membrane form or model was proposed by Singer and Nicholson 1972 fluid mosaic model. According to this, nature of lipid enables, that is, the lipid bilayer enables the lateral movement of proteins within the overall lipid bilayer. Okay, so the nature of the lipid enables the lateral movement. The nature of the lipid enables the lateral movement of proteins within the overall bilayer. There are two different types of protein. That is, the first one, peripheral protein. Second one is the integral protein. With cholesterol is there. The cell membrane has cholesterol, glycolipids, glycoprotein. This is a previous year MCQ. Sometimes you will give the answer as cholesterol, but cholesterol is present in the human or eukaryotic cell membrane. Its function is selectively permeable, helps in the transport of molecules across it. Through different methods are there. It can be an active transport, it can be a passive transport, which we are discussing. So passive transport is the transport from higher to lower concentration. Passive transport means the transport from higher to lower concentration. Higher to lower concentration. It does not utilize energy. For example, diffusion. So passive transport, diffusion is an example for a passive transport, that is a transport of molecules from the higher concentration to the lower concentration, and that does not require any kind of energy. The second thing is the active transport. Active transport means it utilizes energy in the form of ATP, and moreover it is against the concentration gradient, that is from lower to higher. So passive transport is along the concentration gradient. Along the concentration gradient. Active transport is against the concentration gradient. What do you mean by along and against? Along means from higher concentration to lower concentration. Lower concentration. But what is against? Lower concentration to higher concentration. For example, ATP is pump. Just a mention is there in your NCRT, so we mention it already. Is it clear? Next is the cell wall. So I'll highlight it, which will give you a more clear idea about it. Okay, everyone. So next we are going to discuss about the cell wall. Cell wall is a non-living rigid structure, which gives shape to the cell, protection, which also functions as protection to the cell. It helps in cell to cell interaction and provides barrier to undesirable molecule. So that is all the function of the cell wall. Next is the endoplasmic reticulum, or ER. So you already knows what is endoplasmic reticulum. It is a network of tiny tubular structure. Endoplasmic reticulum divides the entire cell into intracellular spaces into two compartments. That is a luminal space, which is inside the endoplasmic reticulum, and the extraluminal space, which is the cytoplasm. So rough endoplasmic reticulum is there, smooth endoplasmic reticulum is there. That is RER, or rough endoplasmic reticulum, is attached to the outer surface. The function is the rough endoplasmic reticulum in protein synthesis and secretion, whereas a smooth endoplasmic reticulum lack ribosome, and it is a site of synthesis of lipids. Is it clear for everyone? So these are the important points which we have to discuss over here. Next is all about the lysosomes. Lysosomes are known as the suicidal bags of the cell. Their function is intracellular digestion. Membrane bound, they are single membrane bound vesicular structures formed by the process of packaging of Golgi apparatus. So the organelle which is formed by the packaging of Golgi apparatus is known as what? Lysosomes. They contain hydrolytes or hydrolyzing enzymes such as lipases, proteases, carbohydrates is there, which are active only in acidic pH. Next is the Golgi apparatus, was discovered by Camillo Golgi in 1898. It consists of small stack-like structures called cisternae, which are packed parallel to each other. They have two phases, that is a convex or the cis or forming phase, concave or the trans or the maturing phase. Important MCQ. Function is it performs the packaging of metals. It has important site. So Golgi bodies are an important site for the formation of glycoproteins and glycolipids. Glycoproteins and glycolipids. Next is the vacuole. Vacuole, you have to understand, tonoplast is a single membrane of the vacuole. They are membrane bound space found in the cytoplasm. For example, if it is a plant cell, consider this is to be a plant cell, guys. So inside the plant cell, what you will actually find? What is actually you are gonna find? You're gonna find nothing. You're gonna find a structure like this. A large space is accommodated by the vacuole, whereas you will see like the nucleus is occupying a small space. Okay, the nucleus is pushed to a side. Okay, I hope you can understand. So guys, this small structure is referred to as the nucleus, and this structure is the vacuole. So inside this you have What the vacuole is covered with the fluid, is covered with fluid, which is known as the cell sap. Okay, contractile vacuole for excretion, contractile vacuole for excretion in amoeba, and food vacuoles are found in protists for the digestion of food. Clear, everyone? I hope it's perfectly clear. Next is the mitochondria. Mitochondria are the Mitochondria are the double membranous structure, and the outer membrane, smooth, and inner membrane forms infoldings, which is known as the cristae. The matrix of mitochondria is known as the inner compartment of the mitochondria. This is like the mitochondria, like, I'll draw an image. Don't worry, we have the tools. Right, everyone? So we have the tools over here. So let's draw. So this is a structure. I'll draw it over here. Okay. A structure like this. Is it? Okay, boundary also. I have drawn a boundary too. Next, please check what I am going to do. It has certain infoldings. Okay, actually, if you check the mitochondria, it is like this. Right, everyone? If the mitochondria is like this infoldings of the mitochondria, you will call them as nothing but the cristae. And the inner compartment, you will call them as the mitochondrial matrix. Right, everybody? Is it clear? Okay, if this was the mitochondria, then the structure would have been like that. Is it clear for everyone? So, mitochondrial function is a site of aerobic. These are all things you know, and I hope you won't never make mistake. That's we're going a bit fast. So, mitochondria is the power site of aerobic respiration. They are called the powerhouse of the cell. And the mitochondria, ribosomes in case of eukaryotic cells are generally 80s ribosome, but inside the organelle, ribosome is an organelle, within an organelle it is 70s inside the mitochondria. It divides. The mitochondria undergoes division by binary fission. Next is the plastid, which is found in the plant cell and euglenoids. Chloroplast, chromoplast, leucoplast are three types depending upon the pigments contained in them. So before we move on to the next one, let me tell you we have made an updated playlist. Okay, guys, we have an updated playlist in our playlist session of Biopoint NEET. Of Biopoint NEET, you will find the playlist session where you can have the session on environmental issues, chemical coordination and integration, then reproduction in organisms, breathing and exchange of gases, sexual reproduction in flowering plants, living world, ecosystem part 1, ecosystem part 2, body fluids, digestion and absorption. Guys, all these things, this is only just 10 videos given over there. Apart from this 10 videos, you have more than 25 plus videos in the updated playlist. So those who are brand new to the channel, or those who have any doubt in any of this chapter, go to the playlist session, search for the video and watch the video available over there. Is it clear? Hearts on the screen, everyone. Let's move on. Oh my god. Chloroplast, green colored plastid is a chloroplast. It contains chlorophyll, double-stranded DNA, 70s ribosome. Trap light energy for photosynthesis. Chromoplasts are the colorless plastid. Carotenoids are fat soluble like carotene, xanthophylls, and others. They give color to the plant parts like fruits and flowers. Next is the leucoplast, amyloplast, eleoplast, and aleuroplast store protein. Amyloplast store starch, eleoplast store oil and fat, aleuroplast store proteins. Ribosomes are composed of RNA and protein without membrane. They are membraneless organelle. Eukaryotic ribosome are 80s, but organelle within an organelle, that is inside the mitochondrial plastid, you will find the 70s ribosome also. They are the site of protein synthesis. I'm not going to explain it everything because you know all these concepts. Ribosome protein synthesis studying right from your 5th standard or 6th standard onwards. So there is no need for a NEET aspirant to be again, for a teacher teaching the NEET aspirant to keep telling all these things again and again and again. I hope there is no need for that. Is it, everyone? Yes, let's move on. That's the cytoskeleton, which is a network of filament, the proteinaceous structure in the cytoplasm made up of microtubules and filament. Cytoskeleton function is a mechanical support, motility, maintenance of the shape of the cell, etc. are the function of the cytoskeletal system of eukaryotes. Cilia and flagella are two important things which may be confused with these centrioles given in the textbook. Okay, everyone. Let me adjust the mic. Yes. So I'll highlight the points. Okay, if you have any doubt, just comment me on the comment section. I'm ready to explain. Okay. Cilia and flagella. Cilia are small structures which work like oars which help in movement. Clear? Flagella are longer structure which are responsible for cell movement. They are covered with a plasma membrane. The core is known as the axoneme, and the cilia or flagella of eukaryotes have a 9 plus 2 array, or prokaryotes. The cilia and flagella. The cilia have a 9 plus 2 arrangement of the axonal microtubules. Next is the centrosome. Centrosome or centrioles are the next topic which we are gonna study. I hope you can hear me properly. My throat is aching, that's why I'm speaking a bit low. If you can hear me properly, just comment me in the comment section so that, or in the chat box. Okay, everyone. So in just next 2 or 3 minutes, I'll increase my voice. Okay, everyone. Okay. Centrosome or centrioles. Next topic. Centrosome contains two cylindrical structures called the centrioles, which is surrounded by an amorphous pericentricular material, centriolar material, made up of 9 evenly spaced peripheral fibrils called the tubulin protein. Centrioles forms the basal body of cilia or flagella and spindle fiber for cell division in animal cell. Okay, everyone, is it clear? Oh my god. Yes. Nucleus. Next structure is the nucleus. Double membranous, perinuclear space and nuclear pores. Nucleoli is the site of RNA synthesis, named after Robert Brown in the year 1839. Chromatin material plus non-histone protein, which is named by Fleming. Nucleoplasm is nucleolus plus chromatin, and nuclear membrane is the perinuclear space with nucleopores. Okay, let me tell you, very important. So we have discussed 3 important points, which is very, 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 very important. Clear, everyone? So let's move on with the next topic. Yes, everyone, is it clear? Chromosome, RNA or DNA plus a histone protein and non-histone protein. Centrosome is a primary constriction. Like, guys, we call it as to be, if we are drawing a chromosome like this. Oh my god. Yes. Perfecto. Right, everyone. I'll manage. Okay, I'll manage. So the small structure in the center, the small structure in the center through which the fibers, all the fibers gets attached during spindle fiber formation. This small structure, you'll call them as what? The centromere. I'll make out a clear image for each one of you. Listen very carefully, everyone. Oh my god, it's not getting clear. Yes, anyway, I have managed to do. So the central structure. Okay, everyone. So the central structure. Okay, this structure I'm telling about. I'll highlight it with you for your kind information. I hope you all know this small structure. You will call them as the small structure. You'll call them as the centromere. So into the centromere, you will find the spindle fibers attached, like the spindle fibers attaches into the centromere. Not from all the sides it will make its attachment. It will make its attachment from two different sides. Right, everyone, it will make its attachment like this. Yes, I hope what I am telling is clear for you. That is, during the mitotic spindle formation, the chromosomes are getting aligned like this, right? So that's what we are focusing or telling over here. So that's what we discussed about the centromere. The primary construction in every chromosome, kinetochore, is a disc-shaped structure on the side of the chromosome, which is very, very important. Okay, everybody, is it clear? So I think we are moving. Okay, everyone. Chromosomes based on the position of centromeres, metameric. Oh my God. Take out the annotation pen itself. I think that would be more better. Metacentric, submetacentric, acrocentric, and telocentric. What is actually metacentric? Middle centromere. Metacentric, middle centromere. Submetacentric, centromere nearer to one end. And acrocentric is centromere situated close to one end. Telocentric, terminal centromere. Very important, repeated previous year question. So that's all about the chapter cell, the basic unit of life. So thank you guys. See you in the next class with some other chapters. Bye bye.